I'm Connie.
I'm Isabel.
I'm Christina.
We're going to be talking about modification of anti-CD20 CAR T cells to target unresectable stage 3-4 melanoma.
This is Connie, and I'll be talking about the introduction. The article looks into what would happen if a modified anti-CD20 CAR T cell was used to target patients with third and fourth unresectable melanoma. A second generation lentiviral vector was used for the production of the CD20 CAR T cell on the Clinic Max prodigy. Now, what is the anti-CD20 CAR T cell? CAR T cell stands for chimeric antigen receptor T cell, which are T cells that have a CAR receptor that target CD20 protein located on B cells. The cells are used to treat different types of cancers. Now, why should we care? This can help decrease mortality rates, can also help provide long-term remission in cancer patients and provide other options of care for patients who have tried other treatments.
This is Isabel, and in the end, the author came to the conclusion that the Clinimax prodigy was successful in manufacturing anti-CD20 CAR T cells that caused a strong targeted immune response against the cancer cells. This proved that the mass production of CAR T cells is foreseen and can also be modified for different targets to reduce patients' treatment time and hopefully minimize chemotherapy treatments. A series of experiments were conducted to prove this theory, starting with a group of patients that consented to testing. This is Isabel talking about Figure 1. The beginning of testing consisted of a total CD3 plus cell expansion count that was measured from each of 9 patients at day 0, day 5, and day 12 of the experiment to determine whether they had enough CD3 plus cells to continue with the manufacturing process. They took blood samples from each patient and used flow cytometry that uses fluorescent dye, specifically eFluor 780, to identify the CD3 plus T cells. They found that each patient had a sufficient amount of CD3 plus cells to continue with the manufacturing process. These cells are a crucial part of constructing CAR T cells. Figure 1b displays the total CAR T cell expansion count that was measured from each patient on day 5 and day 12. The cells are activated and go through genetically modified transduction to express the CAR T cells, which are then placed in a cell culture medium to increase the number of CAR T cells produced. The cell count was measured at day 5 because it is 4 days after transduction, meaning the cells are ready to begin expanding. At day 12, the cells are just finished with expansion, and the final number of cells are measured to determine what the dose of the treatment would be needed for each patient. Most patients had a successful enough expansion to receive the highest dose of treatment, meaning their expansion succeeded as planned. Lastly, Figure 1c demonstrates a pairwise comparison of the expansion of CD3 plus cells and CAR T cells. Each measurement for the unmodified CD3 plus cells are connected to one genetically modified CAR T cell measurement. Each connection represents one production process. While there is no significant difference between the expansion rates, this is important to note because this graph proves that adding CAR genes to the T cells to genetically modify them does not inhibit their ability to grow. This means that there should be no concern for an insufficient amount of CAR T cells being produced for cell therapy.
This is Connie again. In Figure 5 shows how CAR T cells are functioning effectively. CAR T cells were placed with 2 different cancer cells, one of them known as NOM6 cells with low CD expression and JCO1 cells with high CD expression. Panel A shows the activation of the CD20 CAR T cells. The CD20 CAR T cells were activated by stimulation through CD3 and CD28 using agonistic antibodies. The amount of IFNY that was produced through activated cells was measured. Using the ELISA technique, results show an increase in IFNY production compared to the control. Panel B shows cytokine secretion upon target cell stimulation. CD20 CAR T cells were placed with 2 cancer cells, which, when stimulated, CAR T cells produce cytokines, and which were then measured using the Luminex technology. The data shows a production of cytokine when exposed to CD20 positive cancer cells. Expressions of CD20 on the 2 different cancer cells were confirmed by flow of cytometry. In panel C, Expansion of the CD20 CAR T cells in cancer killing demonstrates the expansion of the CD20 CAR T cells after the first stimulation with either low CD20 cancer cell or high CD20 cancer cell. Number of cancer cells was also measured, showing that CAR T cells can reduce cancer cell population.
Hi, this is Christina. So figure 6 overviews the results of using CAR T cells against cancer cells, specifically against NALM6 and GCO1. 4 patients were placed on trial. When we look at figure 6A, we look at the CD107A with regards to GCO1 and NALM6. CD107A is a protein found in the membranes of the lysosome. It plays a key role in cellular cleanup. It is a marker of the cytotoxic activities in immune cells. NALM6 looks at leukemia cells, while GCO1 looks at lymphoma cells. Reason why we use this in the study is because both cells lines provide a different type of CD20 positive cancer cells, leukemia and lymphoma. Figure 6b shows the effects of the concentration of CAR T cells and cancer cells after repetitive treatments. GCO1 shows a definite elimination of cancer cells after the first round of treatment, while the NALM6 shows a variation and an increase of cancer cells after the third round of treatment. For the key experiment, figure 6 clearly demonstrates the effects of modified CAR T cells have on cancer cells, specifically the GCO1, which is associated with the lymphoma cells. This supports the hypothesis of the article that it's possible to create a modified CAR T cell to specifically attack cancer cells. Mass production is successful while having successful results.
This is our references and our acknowledgments for this research article. Thank you for listening to our presentation.
Thank you.